Photosynthesis Required Practical Part 1 by KayScience.com. In this required practical, we're going to investigate the effect of light intensity on the rate of photosynthesis. So we start off with our light source. This here is our light source. You place a ruler away from the light source. So the light is going to pass through the beaker of water. You then get your boiling tube, place your plant specimen inside the boiling tube. The plant specimen is normally Elodea, which is a pondweed. And then you add water to your boiling tube. You then attach a syringe to your boiling tube and make sure it's airtight. So the light is going to pass through the beaker of water. However, the thermal energy is going to be transferred into the water. So that means that the light can be transmitted through the beaker of water to the pondweed, but we can control the temperature because the beaker of water is going to have the heat energy transferred into it. And the pondweed should then start undergoing photosynthesis and releasing oxygen and producing glucose in its chloroplasts. So let's recap. This is the light source. This is the beaker of water where the thermal energy is going to be transferred into. So to control the temperature, so a beaker of water will absorb the heat, but it will let light through. We leave each boiling tube with pondweed in it to run for the same amount of time. So we leave it for the same amount of time. This is another control variable. So we leave it for the same amount of time. This is another control variable. It's question time. Attempt these questions to check your understanding. So now we've set up the equipment, let's look at the science behind it. So photosynthesis is the reaction whereby carbon dioxide reacts with water to form glucose and oxygen. This reaction happens because the chlorophyll and the chloroplasts absorb light energy. The glucose will not be released into the water. What will be released into the water is the oxygen. So the oxygen is going to be released from the plant into the water of the boiling tube, and it is the oxygen that we're going to collect in the syringe. So this is the pondweed that is going to photosynthesize and then produce oxygen that it will release into the water, and the syringe is going to collect that oxygen. That is what we're going to measure. So the volume of oxygen collected by the syringe is going to be our dependent variable. Our dependent variable. It is what we measure. So if you increase the light intensity, so the intensity of the light that the pondweed receives, you're going to increase the rate of photosynthesis up to a certain point. So if you increase the rate of photosynthesis, you're going to increase the volume of oxygen that is being released by the plant. So our dependent variable, what we're measuring, is the volume of oxygen collected inside the syringes. However, what are we changing? What is the independent variable? So what we're changing is the light intensity that the pondweed is receiving. So the way that you change the light intensity is going to be by having the pondweed further away from the light source. The further away from the light source the pondweed is, the lower the light intensity. The closer the pondweed is to the light source, the higher the light intensity. So we've set up 3 test tubes here with the same volume of water in each one, with the same species of pondweed, with the same boiling tube, with the same syringe. These are all control variables. All of them are control variables. We keep these the same, so our results are reliable. The pondweed closest to the light source is going to receive the highest light intensity, and as you go further away from the light source, the light intensity will decrease as shown here. So the light intensity decreases the further away the plant is from the light source. So what we expect to see is the further away the pondweed is from the light source, and therefore the volume of oxygen produced should decrease the further away the plant is from the light source. Because if you decrease the light intensity, you're going to decrease the rate of photosynthesis, Therefore, if there is a lower rate of photosynthesis, there's going to be a lower rate of production of oxygen by the chloroplast in the plant. Therefore, our syringes are going to collect a lower volume of oxygen the further away the plant is from the light source. It's question time. Attempt these questions to check your understanding. So what are our variables? So we need to know our independent variable, our dependent variable, and then all the control variables. Our independent variable was changing the light intensity by changing the distance the pondweed was away from the light source. The dependent variable was measuring the volume of oxygen that was released by the pondweed. So we measured the volume of oxygen. So we controlled the temperature, we controlled the carbon dioxide concentration, we controlled the time that the Eladia was left in the boiling tube to photosynthesize. We also controlled the pH, and we also controlled the plant that we used in each boiling tube. So we used pondweed, Eladia in each test tube. It's also important to control the size of the pondweed that you use, because if you use different size pondweeds, the larger pondweed will release more oxygen. It's question time. Attempt these questions to check your understanding. Press pause to practice using those key words. The answers will follow. Press pause to go through your answers and make any corrections to your mistakes. Press pause to answer the questions. The answers will follow. And if you're stuck, just rewatch the video. Press pause to go through your answers and make any corrections to your mistakes. Visit KayScience.com for more free videos, worksheets and quizzes at KayScience.com. And don't forget to like and subscribe.